Hello, my dear students. Welcome to our channel BioRealm. Up till now, we have seen what is cell. Then we talked about cell theory. Then we have seen the overview of the cell. Now, in this video, we'll talk about the prokaryotic cell. In overview of the cell, we talked briefly about what is prokaryotic cell and what is eukaryotic cell. But in this video, we will go in more detail of what exactly the prokaryotic cell is about, what characteristics that prokaryotic cell possesses, what kind of structures that prokaryotic cell are having. So this video will be more interesting and it is in more detailed form. We'll see minute things regarding the prokaryotic cell. So without further delay, we'll start with prokaryotic cell. Okay. So, first, I have to discuss some basic things about prokaryotic cell, and after that, we'll talk about or we will discuss the prokaryotic cell in detail. So, in nature, if you see prokaryotic cell, it is represented by certain organisms. That organisms are, write, let me write, that organisms are bacteria, then blue, green algae, which is sometimes called as cyanobacteria. Then third is your mycoplasma. So what are mycoplasma actually? Mycoplasma, these are the smallest kind of bacteria, smallest bacteria. And one peculiar characteristic of this mycoplasma is they are not having any cell wall, means cell wall is absent, cell wall is absent. And the fourth one is given in your NCERT textbook is PPLO, that is pneumonia like organism. So what are these PPLO? It is nothing but the mycoplasma. Always remember PPLO is as same as that of the mycoplasma. Certain mycoplasma which are present in the pleural, you can say pleural cavity of certain cattles which are suffering from pneumonia. In such pleural cavity, these organisms, these mycoplasma occur, and therefore these are previously called as PPLO, that is pleuropneumonia like organisms. Means they are infectious organisms. PPLO are the infectious organisms. They are the mycoplasmas. And if we see the characteristic of mycoplasma, then mycoplasma are the bacteria which are smallest among the bacteria. They are way far small than the normal bacteria, 1,000 times smaller than that of the normal bacteria. Then these are not having any kind of cell wall. So cell membrane is absent. Okay, so that is what about cell wall is absent in mycoplasma. So these are some of the uh, prokaryotic structures which are present in the nature. So what are these prokaryotic structures? First one is your bacteria, blue green algae, mycoplasma, and PPLO, which is nothing but the mycoplasma. So if we talk about the characteristics of uh, bacterial or prokaryotic cell, they are very much smaller in size. They are very much in smaller in size. Okay, they are very smaller in size. Okay, and they carry out variety of function. They have the tremendous variation in their shape. Okay, there is a variation variation in the shape, right? Now, what are the different types of shapes of the bacteria that we will talk? Shapes of the bacteria or shapes of the prokaryotes, you can say. So, first is your bacillus. Bacillus. So, bacillus type of bacteria means what? Bacillus type of bacteria means rod shaped bacteria. Bacillus type bacteria means what? Rod shape. Bacillus means rod shape. Means if the bacteria is having elongated structure like this, like that of the rod, then that type of bacteria is called as bacillus. Okay. So the second one is your second type of bacterial shape is your coccus. Coccus. Okay. So what do you mean by coccus type of shape of the bacteria or this coccus type of shape in bacteria? You will see it is the spherical type, means circular, spherical, oval type of bacteria. Again, it is subdivided into monococcus, okay, then diplococcus, streptococcus, and staphylococcus, okay, and staphylococcus. So, what is this actually? What is monococcus? If the one coccus is present, then it is called as monococcus, okay. Two cocci or two coccus means two spherical structures are present, then it is called as diplococcus. Then if the coccus are present or the cocci are present in chain, then it is called as streptococcus. It is called as streptococcus. And last one, if it is present in cluster, if the cocci means that circular structured bacteria they are present in cluster, then it is called as staphylococcus. Staphylococcus. Okay. So these are what some of the subtypes, but for in your book it is mentioned up to this, so you can memorize. This is for additional information. Okay. Then third type of shape is your. Let me take the one first. Third type Vibrio. Vibrio means what? Vibrio means comma like. It is like it is uh, take the black comma means like this, like this. This is called as comma like. Okay. And last one is spirillum. Last one is spirillum. It is the spiral shaped type of the chloroplast. Spiral shape. So these are four different types of the uh, not chloroplast bacteria, which are the examples of prokaryotic cell. So up till now we have seen what are different prokaryotes. In that we have seen that bacteria are there, blue green algae are there, mycoplasma or pplo are there. Then we talked about its versatility in the shape. We talked that bacteria might be bacillus type, means rod shape. Coccus means spherical or oval. Vibrio means comma like. And fourth one is spirillum means spiral. So now onwards, we will going to see the structure of prokaryotic cell in detail. So let us go from outside to inside. This is the diagram that I have drawn. That I have here. I have showed here, not drawn. I have showed here. Let us discuss from the outside. Uh, Let me take. So if we see the outermost layer, then you will find here this structure. This structure is called as your cell wall. This structure is called as your cell wall. See, we have seen that in overview of the cell. that cell wall is present in case of prokaryotes, whereas, one second, cell wall is present in prokaryotes, and it is absent in case of eukaryotes. Cell wall is present in prokaryotes and it is absent, so it is absent in eukaryotes. Okay, cell wall is absent, not particularly in eukaryotes, but in case of animal cell, the cell wall is absent. Okay, and it is present in plant cell, but it is absent in the animal cell. Okay, but in case of prokaryotes, we have seen that cell wall is present, and in this case, this cell wall is made up of a structure which is called as peptidoglycan. Generally, in most of the case, bacteria which are prokaryotes, the cell wall is made up of peptidoglycan. Peptidoglycan. If we see the structure now, name, it is made up of two different words, peptido and second one is glycan. So peptido means what? It is concerned with the amino acid, because we know that peptide chain is a protein chain, and you know that proteins are made up of amino acid. So it is amino acid, and glycan is your sugar. So these two compounds make the cell wall of the most of the bacteria, right? So uh, this bacteria is having this structure which is called as the cell wall. Then below cell wall, going inside, we are going inside, and below the cell wall, we will see the outermost limiting membrane, and that membrane is nothing but, that membrane is called as the cell membrane. This membrane, if you see below the cell wall, you will see a membrane. You can see here also. This membrane below the cell wall is called as your cell membrane. Cell membrane. Okay, so it is present below the cell wall. Now inside to that of the cell membrane, you will find fluidic matrix or fluid like structure. Fluid matrix, you can see, I am drawing this, this black dotted structure. It is a fluid matrix structure which is called as cytoplasm. This is called as the cyto is called as the cytoplasm. This is called as cytoplasm. It is the fluid matrix. Okay. Now we talked about prokaryotic cell in overview of the cell, that it is not having the membrane bound nucleus. Means nucleus is almost absent. Pro means what? Primitive. Karyon means what? Nucleus. Primitive nucleus means what? Some, in some bacteria or in some prokaryotic, nucleus may be present, but that is not having any membrane. Means if you see the chromosome, the bacterial chromosome, the bacterial DNA in the center, this is what the bacterial chromosome is there, and this chromosome or DNA, this one is your bacterial DNA. This is the bacterial DNA or chromosome. This bacterial DNA or chromosome, it is called as the naked one. Naked one. Why it is called as naked? Because here it is not surrounded by any membrane bound structure, which is called as nucleus. Means it is freely suspended in the cytoplasm. ये जो है बेटा, ये डीएनए जो होता है बैक्टीरिया का, वो फ्रीली साइटोप्लाज्म में सस्पेंडेड होता है. इट इज नॉट कवर्ड विथ any sort of membrane, any sort of membrane bound structure which is present in eukaryotic cell. In eukaryotic cell हम लोग देखते हैं कि सेंट्रली एक न्यूक्लियस होता है, उसके अंदर डीएनए या क्रोमोसोम्स जो होते हैं वो उसके अंदर प्रेजेंट होते हैं, लेकिन बाहर से मेम्ब्रेन बाउंड स्ट्रक्चर होता है जिसे हम न्यूक्लियस कहते हैं इन केस ऑफ यूकैरियोट्स, बट सच स्ट्रक्चर इज एब्सेंट इन केस ऑफ प्रोकैरियोट्स। प्रोकैरियोट्स में जो होता है बेटा कि प्रोकैरियोट्स में न्यूक्लियर मेम्ब्रेन ही नहीं होता है, तो बैक्टीरियल जो डीएनए होता है वो फ्रीली सस्पेंडेड होता है साइटोप्लाज्म में। ओके, नाउ अपार्ट फ्रॉम दिस bacterial DNA, certain extra chromosomal DNA, okay, certain, let me take another color, extra chromosomal circular, generally circular DNA are present in the bacteria which are called as plasmids. Plasmids, okay, and these are the plasmids you can see here. So this circular structure which is present, this is the bacterial chromosome, but apart from this bacterial chromosome, extra chromosomal means it is present outside that bacterial DNA. Another extra chromosomal circular, generally it is a circular form of DNA which is present, that is called as plasmid. So what is the function of plasmid? It provides certain unique phenotypic characters, phenotypic phenotypic characters, यूनिक फिनोटिपिक कैरेक्टर्स, मतलब कि इस पर भी कोई जीन्स प्रेजेंट होते हैं, सर्टन सम अमाउंट ऑफ जीन्स, some genes are present on plasmids also. No doubt bacterial DNA is having most of the genes, but in plasmids certain genes are also present, and these genes are there which possesses certain characters. For example, antibiotic resistance, antibiotic resistance, it is the characteristics which is present in the plasmid DNA, or the gene for that is present in the plasmid DNA. Apart from that, these plasmids can carry out the bacterial transformation with फॉरेन डीएनए, मतलब कि इसमें कुछ जो जीन्स होते हैं इनमें ट्रांसफॉर्मेशन की एबिलिटी होती है, मतलब कि ये प्लाज्मिड के जो जीन्स होते हैं वो दूसरे ऑर्गेनिजम्स में भी इन्फेक्ट कर सकते हैं, उनमें जाके इंसर्ट हो सकते हैं। तो अगर हमें यहाँ पर इस प्लाज्मिड के जीन में हम लोग अगर कोई भी डिजायर्ड डीएनए जो कि फॉरेन डीएनए है वो इंसर्ट करेंगे, तो इस प्लाज्मिड के डीएनए के साथ वो जो हमने इंसर्ट किया हुआ डीएनए होता है, वो फॉरेन डीएनए, वो भी दूसरी जगह हम ट्रांसफर कर सकते हैं, और इसके वजह से बैक्टीरियल ट्रांसफॉर्मेशन हो सकता है। इसका जो यूज है वो जीन ट्रांसफर ट्रांसजेनिक एक ब्रांच होता है जिसमें जीन ट्रांसफर टेक्नोलॉजी, जेनेटिक इंजीनियरिंग जिसे कहते हैं हम लोग, तो उसमें इसका बहुत सारा यूज किया जा रहा है। इस ये हम अलग टॉपिक में इसे डिस्कस करेंगे। ओके, तो हमने यहाँ पर क्या क्या देखा? अप टिल नाउ वी हैव सीन दैट इट इज हैविंग सेल वॉल व्हिच इज मेड अप ऑफ पेप्टिडोग्लाइकॉन, below cell wall cell membrane is present, or which is called as plasma membrane. Then below plasma membrane fluid matrix is present which is called as cytoplasm. In cytoplasm the DNA is freely suspended, bacterial chromosome or bacterial DNA, or it is also called as a nucleoid. It is also called as nucleoid. Okay, and it is naked DNA. Now, as we have seen in eukaryotic cells, eukaryotic cell possesses certain membrane bound organelles which are absent in case of prokaryotic cell. We have seen in eukaryotic cell, in overview of the cell, that mitochondria, chloroplast in case of plant cells, endoplasmic reticulum, Golgi bodies, lysosome in case of animal cells. So these are the membrane bound organelles which are present in the eukaryotic cells, but which are absent in case of prokaryotes. But one cell organelle which is not membrane bound, that occur in prokaryotes. Okay, that non-membrane bound organelle is nothing but the ribosome. That this is the ribosome, which is non-membrane bound organelle. Okay. So this is present in prokaryotes, and it is having the type of 70S. S stands for Swedberg unit, which is used to represent this ribosome. That we will see in detail in when we study the protein synthesis. In that case, I will explain you what is S and what is 70 there. Okay. So for our time being, just memorize this, that ribosomes, which are non-membrane bound organelles, they are freely present in the cytoplasm. These are only organelles which are present in prokaryotic cell. Okay. Now, apart from that, this structure, cell-like structure, which is called as flagella, flagella, or singular is called as flagellum. It is important in the movement of the bacteria, or locomotion, means bacterial locomotion or movement takes place with the help of flagella. Now there are certain structure which are called as pili. These are called as pili. So these pili like structure, these are very important in the movement of the bacteria, मतलब कि बैक्टीरिया के मूवमेंट के लिए, प्लस एडेजन, एडेजन जब भी बैक्टीरिया कहाँ पर भी इन्फेक्ट करता है या तो फिर अनदर वायरस बैक्टीरिया को इन्फेक्ट करता है, तो जो एडेजन का प्रोसेस होता है, मतलब कि जो बॉन्डिंग होता है, जिसके जिसके वजह से जो है वो जो प्लाज्मिड डीएनए होगा या तो फिर बैक्टीरियल डीएनए होगा, वो ट्रांसफर होता है एक ऑर्गेनिज्म से दूसरे ऑर्गेनिज्म में, उसके लिए जो है वो पिलाई जो है वो हेल्प करते हैं। इतना ही ध्यान में रखिए, एडेजन मीन्स अटैचमेंट प्लस मूवमेंट, ये जो फंक्शन है ये पिलाई का फंक्शन है, वेरस फ्लैजेलम इज मेनली रिस्पॉन्सिबल फॉर मूवमेंट। outermost layer outermost covering is also there, which is called as the capsule. Capsule is also present, which is also again made up of sugar structure. Okay. So, and we forgot to talk about the bacterial plasma membrane. Here we talked about cell membrane or plasma membrane, but it is having certain intrusions into the cytoplasm. It is not drawn here, but here you will find such intrusions of cell membrane into the cytoplasm. So these are called as mesosomes. Mesosomes. Okay, these are called as mesosomes. Okay, so these are the intrusions of the plasma membrane. Specially differentiated cell membrane or plasma membrane is present in the prokaryotes, which is called as mesosomes. Okay, so these mesosomes help in the process of your protein synthesis. Is there certain functions are assigned, or that functions get performed because of these intrusions. Okay, so I hope you have understood the overall structure of prokaryote. In brief, let us summarize this. We talked about prokaryotes. It is represented by bacteria, blue-green algae, which are called as cyanobacteria, mycoplasma, then PPLO, which are nothing but the mycoplasma. Then we talked about different shapes of the prokaryotes, particularly bacteria. In that we see about bacillus means rod shape, coccus means spherical, then We have seen Vibrio means comma-like and spirillum means spiral. When we talked about the cell wall in bacteria, it is made up of peptidoglycan. Below cell wall, cell membrane is present, which is a specialized type of cell membrane, which is a differentiated cell membrane, that particularly called as Misosome. It has to perform certain functions. Fluid matrix in the center, which is present, which is called as cytoplasm. DNA, naked DNA more precisely, is present freely suspended in the cytoplasm. It is not having any nuclear envelope. Membrane bound nucleus is absent, therefore it is a prokaryotic cell. Then cytoplasm contains non-membrane bound organelle, which is called as ribosome. The ribosome function of ribosome is very important in case of protein synthesis. Then apart from this bacterial DNA, extra chromosomal circular DNAs are also present, which are called as plasmids. That plasmids are very important in case of or they are having certain genes which control certain phenotypic characters like disease resistance, antibiotic resistance. They also responsible in the bacterial transformation. We will talk in the genetic engineering or transgenics. Then we talked about flagella, mainly responsible for movement, villi, adhesion, and movement. So I hope you got the detailed structure of prokaryote. And if you have any doubt or if you like the video, if you watch the video, then please like the video and give the comments in the comment sections. And if you like the video, please share it to the students who are preparing for the NEET. Till then, thank you.